Vanakkam, good morning. என் பெயர் டாக்டர் பிரபாகர். Today we are going to discuss about the excitation contraction coupling or note on muscle relaxation. This is the important frequently asked university long question. This video will helpful to get the concept about the excitation and contraction coupling. You must read the textbook, that will helpful to you. So we will go to the topic. What is excitation contraction coupling? Excitation of the muscle fiber causes development of action potential, which through sequence of events produces muscle contraction. This is the excitation contraction coupling. The initiation of the excitation contraction coupling is done by the calcium ion. So calcium ion is the coupling agent for the excitation and contraction. Excitation contraction coupling events occurring between the stimulation of the motor nerve and muscle contractions. The stimulation of the motor nerve, release of acetylcholine from the presynaptic membrane of the neuromuscular junction. The neuromuscular junction contains presynaptic membrane and postsynaptic membrane. Presynaptic membrane is mainly made up of nerve. Postsynaptic membrane is the muscle. This area is the synaptic cleft. The acetylcholine is released from the presynaptic membrane. It binds with the acetylcholine receptor located in the postsynaptic membrane. So bind with the acetylcholine receptor located in the postsynaptic membrane. It produces the end plate potential here, the muscle cell membrane, sarcolemma, end plate potential. So it increases the sodium permeability in the sarcolemma. So it produces the action potential. It is travel along the T-tubule. The sarcolemma contains the invagination that produces the transverse tubule. The action potential travel along the action potential. So we will see the following events in the next slide. This diagram shows the sarcotubular system. Sarcotubular system is the modified endoplasmic reticulum. Main function of the sarcotubular system is the storage of the calcium ions. The sarcotubular system contains one T tubule and two L tubule. T tubule is the transverse tubule, L tubule is the longitudinal tubule. T tubule contains dihydropyridine receptor, that is the voltage sensitive calcium channels, and L tubule contains terminal systems of the L tubule contains ryanodine receptors, that is ligand cutter calcium channels. Both are physically dihydropyridine receptors and the ryanodine receptor. Both are connected physically and mechanically through the locking protein at rest. When the action potential is traveled through the T tubule, the structural changes occur in the dihydropyridine receptor. It opens the ryanodine receptors. It releases the calcium ion. It increases the cytoplasmic calcium level 2000 times from the 10 power minus 7 moles per liter to 2 into 10 power minus 4 moles per liter. That is important for the initiation of the excitation contraction coupling. The another one, calcium pump, is present in the longitudinal tubule, that is called as the SERCA pump, sarcoplasmic endoplasmic reticulum calcium channel. That is important for the muscle relaxation. This picture shows the myosin binding site of actin at rest and binding of troponin with calcium ions. At rest, myosin binding site of actin is covered by the tropomyosin complex. The blue color circle is the tropomyosin. Troponin I complex covers the myosin binding site of actin. When the calcium ion bind with the troponin C, the tropomyosin move away from the myosin binding site of actin. Important events of excitation, contraction, coupling. Muscle at rest, tropomyosin, the blue color line, and troponin I, small orange circles, covers the myosin binding site of the actin. When the calcium is bind with the troponin C, it uncovers the 7 myosin binding site of the actin, and myosin is bind with the actin. And with the use of high energy phosphates, pull the thin filament towards the center of the sarcomere. It uses the high energy phosphates. For the detachment of the myosin head from the actin, the ATP is attached to the myosin head. Same time, calcium is pumped back into the sarcotubular system through the sarcoplasmic endoplasmic reticulum calcium pump, CERCA pumps. That is important for the muscle relaxation. Inside the cytoplasm, the calcium ion concentration is decreased 10 power minus 7 moles per liter. And the myosin head has the myosin ATPase activity that hydrolyzes the ATP to ADP and high energy phosphate. Again, muscle contraction occur. Summary of the excitation contraction coupling. When the motor nerve is stimulated, releases the acetylcholine from the presynaptic membrane, bind with the acetylcholine receptor on the postsynaptic membrane, produces the end plate potential. When it reaches the axon potentials, travel through the T tubule and stimulate the digateropyridine receptor. It produces the structural changes. It opens the calcium from the ryanodine receptors. Inside the cytoplasm, calcium ion concentration increases. The calcium bind with the troponin C. Each troponin C bind with the calcium ions, uncover the 7 myosin binding site of the actin. Myosin head is bind with the actin, and with the use of high energy phosphate, pull the thin filament towards the center of the sarcomere. That is called as the power stroke. And ATP is detached from the myosin head with the help of ATP attachment. The calcium level in the cytoplasm is decreased. It pumped back into the sarcoplasmic reticulum through the CERCA, sarcoplasmic endoplasmic reticulum calcium pump. And myosin head has the myosin ATPase activity. It converts the ATP to ADP and high energy phosphate. Again, muscle contraction occurs. Thank you for watching this video. If you like this video, share your friends. Those who are not subscribed this channel, please subscribe. Thank you once again.